Okay, so in the previous lecture, we talked about basic experiments, and we talked about the importance of the independent variable, the dependent variable, confounding variables, and so forth, and the various designs. But in this particular lecture, I'm going to talk more about the practical aspects of conducting an experiment. So the general or overall learning objective is to cover the practical aspects of design and conducting research, to explain the difference between a straightforward versus staged manipulation, talk about different forms of measurement on the dependent variable, to talk about sensitivity and how you assess floor effects and ceiling effects and how they're problematic for research, and talk about pilot studies and manipulation checks. And by the end of that, we should be done. So, as you may recall from lecture 7, most research operates with samples. And a sample, by definition, is a subset of the population, whereas the population is all parties of interest. So when we do sampling, there are different forms of sampling techniques. There's probability sampling, which the likelihood of being selected is known to the researcher. And then there's non-probability sampling, where the likelihood of being selected is an unknown to the researcher. And when you want to generalize, we talked about in lecture 8 about internal validity, but we also should talk about external validity. So external validity is the ability to generalize out. You want to have probability sampling, because knowing the likelihood of being selected is important to knowing the sampling frame, and that's quite useful. However, much research is done with non-probability sampling. Just convenience sampling is the most common way that people do research. And what is convenience sampling? It's a form of non-probability sampling where you just take whomever volunteers. And when we think about that, non-probability sampling, it's not necessarily a threat to internal validity, but it does affect external validity or generalizability. So long story short, if you want to talk about the population as a whole, you need to have probability sampling. Right now, we're going through the election, 2020 election. And to me, I think that's a good example. How do they know when to call a state? And I'm not saying that every state that has been called is actually the final vote total and what will be, but typically they know how many registered voters there are. They know how many outstanding ballots there are. They know the form of voting that the person requested, whether it be in person or a mail-in vote. And they could take from that which that they have and say, you know what? The odds are that this particular state is going to lean or go to a given candidate. That only works when you know the total number of votes you're expecting in a given state. And so that's an example of probability sampling. Non-probability sampling, if we're going to take the election as an example, would be exit polls. When you do exit polls or polling, many polls have been incorrect in regards to the Senate. There was a belief that the Senate would flip during this election season, and it doesn't look like that's going to happen. There was also a belief that the House of Representatives the Democratic majority would increase even further, and it doesn't look like that's going to happen. So long story short, when you actually are counting votes within a state, that's more consistent with probability sampling. When you're doing exit polls or these polling, you're relying more on volunteers. So that's a non-probability sampling, and it doesn't always generalize well to the population. All right. The next thing when designing an experiment you want to think about is sample size. And the sample size matters up to a certain point. And I believe we said in lecture 7 that as sample size increases, the more generalizable it will be to the population as a whole. But it plateaus at roughly 1,000. So if you go back to lecture 7 slideshow, there was this graphic with confidence intervals and how many participants you needed. So it kind of plateaued at about 1,000. So in general, larger samples are more generalizable to the population than smaller samples, insofar as that they're also representative samples. So if the sample is large but homogeneous, it may not generalize out to the population as much. So you want to pay attention to sample size, but how do you know what the appropriate sample size is for your research? Well, there are sample size calculators that will tell you the appropriate sample that you need. But if you don't know, one tool that's recommended to new researchers is to look at previous research studies and see, well, what was considered an adequate sample size in past research? And that will give you guidance. And the other thing I will tell you is, depending on the kind of research you're doing, the sample size will also differ. So if you're doing vision research and you're using an animal model, some of the brilliant work on PET and SPECT scans and vision were done with the chimpanzee. So you're not going to have hundreds of chimpanzees to do research on. You might have a sample size of 10 that you follow through for a long period of time, versus survey research, which you might have several hundred participants. So you really have to think about what kind of research am I doing, and would this sample size accomplish the goals that I'm trying to do? So after you have figured out your sampling technique and your sampling size, now you have to define your variables. And in lecture 4, we talked about the importance of operational definitions. Now, just to be clear, an operational definition is a specific way that you measure or manipulate a variable. So you're telling the reader how you're going to manipulate the independent variable, or you're telling the reader how you're going to measure the effect of your manipulation on the dependent variable. But that is what we call an operational definition. So first, you figure out how you're defining things. Then you go through your IRB process, which is getting research approval, and then you provide participants with informed consent. Now, in lecture 3, we talked about ethics and the importance of informed consent. It's not enough for a participant to say, yeah, I'll be part of the study. They have to be told reasonable risks and benefits to this study, and potential harm that could occur as a function of this study. In addition, we typically tell the person, well, here is the general theme of what I'm studying. So you tell the reader that, so that they have enough information to decide whether they want to participate. Now, sometimes telling a person what you're going to do could tip off your hypothesis. And if that does, it could skew your results. If you're worried about that, you can use deception, which is kind of to somewhat mislead or withhold information about the study to protect the validity of the results. Now, you may recall, if you use deception, you must use debriefing, which is a process of telling the reader the true nature of the study. Okay. So there are two types of manipulations, as I said in the review. There are straightforward manipulations and staged manipulations. A straightforward manipulation is the kind of manipulation where you manipulate either the wording, you manipulate something about the appearance of the study, or something about the writing or visual aesthetics. But all in all, you're not deceiving the person about what you're doing. So let's take a look at a study that used straightforward manipulation. So LaBranche did a research study exploring health promotion brochures in a health center, and they wanted to see if brochures that gave guidance on doing breast self-examinations, whether that affected one's confidence in doing a self-exam. And in group one, they had a brochure, but it only included text. So it explained how to do a self-examination, but no pictures. And in group two, they had the same text, but what did they manipulate? They added pictures as well, or diagrams as well. And they asked people in one of these two groups, or both of these groups, pardon me, to rate their level of confidence. That's your dependent variable. And what was fascinating was that one's personal comfort with sexual material, and this wasn't overly graphic, it was just showing how to test for lumps in the breast. They found that people who were uncomfortable with sexual material tended to rate themselves as less competent when the brochure included pictures. That's counterintuitive. Versus people who were comfortable with sexual content in general, they didn't have a difference whether it was text only or text plus pictures, which is quite fascinating. Another example of a straightforward manipulation is the Bornstein study in 1998, where they studied the effect of severity of injury on product liability. So the independent variable is severity of injury. So you had low severity versus high severity. And then the dependent variable was what percentage of the people held the company liable. So let's go through the two groups again. So in the low severity group, they took birth control pills, or this is at least the narrative. So a person took birth control pills, and as a function of that, they developed cancer. But when they developed cancer, it was detected early. They only lost, and I put only in quotes, they only lost one ovary, so they could still have children. And in terms of their prognosis, the likelihood of getting better and beating cancer was fairly good. So that's the low severity group. Then you had the high severity. So notice the description, all they changed was cancer was now detected late. They changed the number of ovaries removed. So both ovaries were removed, which interfered with the ability to have children and created a poorer prognosis. So the evidence as to whether the pills were responsible for the cancer was all the same. So all they did was impact the profile of the individual and how they were affected, not the data supporting guilt or innocence for the company. And what they found, which is quite profound, is that 40% of people held the pill company liable in the high severity condition, whereas 21% of people held the pill company liable in the low severity condition. What's the point? The point is, in forensic psychology, in the court system, we don't just pay attention to the evidence, but we pay attention to the damages as well. And if you're on if you're a juror in a case and you have a situation where there is a significant damage, that might supersede the evidence in holding the company liable. So we're aware of this. So obviously there are people who do cross-examinations to challenge things like that. Another example of straightforward manipulations is Petty, Cacioppo, and Goldman wanted to see whether or not the credibility of a person influenced one's attitudes, or whether or not personal involvement impacted one's attitudes about something. So here's the design. College seniors were given a passage and they were asked to read reasons why taking a comprehensive examination should be required before graduating with a bachelor's degree. What is a comprehensive examination? It's a test of your knowledge of all the things you've learned throughout your undergraduate career and the things that the college would expect of you. Now, they manipulated two things. One, the credibility of the writer of the essay. So some people were told that this essay was written by a Princeton University professor, which is an Ivy League university. And then other people were told that this was just a local college professor at a junior college. So there is high credibility versus low credibility, right? Or lower credibility, not to say that junior college professors have low credibility. And then there was a level of involvement. If this survey, the results supported the comprehensive examination, people were told that either it would take place effective immediately, which would affect the people filling out the survey, or it would take place in 10 years, which would not affect the people filling out the survey. And what they found was when the respondent, the person filling out the survey, was not personally involved, if the comps were going to occur 10 years later, then you were more objective and you paid attention to credibility. So you're going to have more attitudinal shifts with a Princeton professor versus a local college professor. However, if it affected you personally, credibility didn't matter. It could be Moses coming down from the mountaintops, and you would oppose comprehensive examinations. And why is that? Because it would affect you. So it's interesting when people weigh in on topics, there are a lot of factors that drive or shape their perspective or attitudes. And personal involvement is one. It's harder to be objective when you're personally involved. So what did those three studies have in common? The thing that they had in common was that the previous three slides were straightforward manipulations. They didn't manipulate anything other than the wording, the text, or they didn't manipulate anything other than the pictures. So it was just very basic manipulations. Whereas now we'll talk about stage manipulations. Stage manipulations require some level of deception or acting on part of the researchers. So one example of this is to use confederates. What is a confederate? A confederate is a person that works alongside the researcher to create a certain experience. So they're actors. They're not actually participating in a study. Now, you might think they're participating in the study, but they're not. So when we think of confederates, let's go back to the Milgram shock experiment from lecture 3. Well, Milgram used an actor by the name of, quote, Mr. Wallace, who was the recipient of the shocks. Now, you thought Mr. Wallace was just another person who was volunteering to be part of the study, but it was rigged so that you were the teacher and Mr. Wallace was always the learner. So that's an example of a confederate. So anytime there is deception or misleading, or actors to kind of create some psychological state or to mirror what happens in the real world, we call that using a confederate. Another example of this was with Solomon Asch, and Solomon Asch was known for the line matching task, right? So he gave you a line and then he gave you a bunch of other lines and he asked you to say, well, which of these 3 lines went with the standard line? But it was staged. It was staged so that you were number 6 in the series and the previous 5 people worked with Solomon Asch and they intentionally gave the wrong answer and they wanted to see whether you would conform to the answer. So that's another example of using confederates or stage manipulations. Here's another example. Fazio looked at the effect of cognition when a person has undivided attention versus split attention. So group 1 was given 10 minutes to proofread a manuscript. Group 2 was also given 10 minutes to proofread a manuscript, but from time to time they were interrupted by the experimenter to do other things. Well, that's a stage manipulation now, right? Because now the experimenter is doing something other than collecting data on cognition. He or she or they are now also participating and getting you to do other things. So why would Fazio want to do that? Well, Fazio knew that when you're in the workforce, there are times that you're going to have the ability to focus on something with pure undivided attention. And then there are going to be other times where you're going to be pulled in multiple directions all at the same time, and you're expected to perform a task. And he wanted to figure out, well, how would you perform? Well, what he found was that obviously when you have split attention or when you're trying to multitask, you actually perform poorer than when you focus on one thing at a time. So I defined what a confederate was. That's a person who's an actor, an accomplice, pretending to be part of the experiment when they're actually working with the researcher. I talked to you about Solomon Asch's study already, the line matching, and what he found was that people, even when they know the answer is wrong, tend to conform if there's a lot of pressure on them. All right, so now these were all done in the lab. Barron did a study in a shopping mall, which is mimicking the real world, which is called a field experiment, in which he had a confederate ask for change when people were busy, and he wanted to see if you would stop and help. So that was that study. Now here's the line matching task. the standard line versus a comparison line, if I were to ask you, well, which comparison line matches the standard line, most of you would say number 2, right? Unless you had some kind of visual problem. Most people asked the question alone would say number 2. But now, if you had 5 people before you say number 3, and nobody said anything other than number 3, you might actually pick number 3, demonstrating conformity, even though you knew it was wrong. And in Solomon Asch's study, they were asked, well, why? Why did you conform? And there are 2 common answers. One was, I didn't want to look like an idiot. So if everyone's saying one thing, I don't want to be the person who says something different. I didn't want to stand on my own. So they conformed. Or they said, you know what, maybe I have poor vision and my eyes were failing me. So if a person like myself who wears contacts was looking at that, I might think that there's something wrong with my contacts. So then you have conformity. Now, what was interesting in Solomon Asch's study was that if you had even one person break ranks, conformity went virtually to 0, which meant or demonstrated that people were picking the wrong answer. Many times they knew it was the wrong answer, but because of these 2 factors I mentioned, they didn't want to look foolish. Okay. So we talked about straightforward manipulation and stage manipulations. When we think of stage manipulations, one of the things that you have to consider is it requires acting ability. It requires a lot more effort and creativity. So it's challenging. And then if other people want to replicate your research, it can be hard for people to replicate your research because they're not you. And perhaps when they do the same things you did, it doesn't draw the same outcome. Whereas straightforward manipulations is very easy to replicate, very easy to interpret, and that's that. So before I shift gears, I will say to you, 9 out of every 10 studies are straightforward manipulations. Only about 1 out of every 10 studies uses stage manipulations, partially because of the concerns we have on this slide. So the next thing is you want to manipulate the independent variable. The first thing you want to think about is the strength of your manipulation. And ideally, you want to have the maximal difference between the 2 levels of the independent variable as possible. And the rationale for that is if you have 2 extremely different groups and there is something to be found there, or there are significant findings, you're going to have more sensitivity when they're worlds apart than if they're closer. Now, you want to maximize the difference. There are 2 exceptions to this rule. If you create such a difference that it doesn't occur in the real world, then you're losing your generalizability, you're losing your external validity, so you might not want to maximize the difference in this case. The second scenario would be as if you're going to cause extreme fear or anxiety. That might be unethical to cause that level of psychological distress. But in general, when trying to develop sensitivity, you start with a very strong difference and then you work your way inward. So to give you an example, let's say we wanted to see romantic relationships, and we wanted to see if people who are more similar or less similar tended to work out in relationships. Well, the first thing you might want to do is do a measure on their level of similarity. And then you would want to correlate how much they liked one another. But you would start with the extremes, let's say 1 and 10, that is very dissimilar from one another. And if you found significant differences, then you would move inward to say 4 and 7, which are far less similar from one another. And you should be able to still identify significant differences, but the mean differences are likely going to be smaller. Now, another thing, this is actually true. This research suggests that the more similar you are to another person, the better the outcomes, and the more likely you're to develop attraction towards them than if they're different. So some of you might have heard the cliche, opposites attract. That is only true with magnets. When it comes to romantic relationships, the more similar you are on the big ticket items, the better it is. Okay. So that was the strength of the manipulation. And next thing you want to think about, and again, lecture 9 is all the practical aspects of designing a study. The cost of the manipulation. So the kind of study you are going to do will depend on your monetary resources or your budget. So if you are very limited in your budget, then you probably want to do things like surveys because it's cheap and it's quick, as we mentioned in lecture 7. Whereas stage manipulations and all of that is going to cost you more money because you have to hire the actor. Similarly, if you have fancy equipment that measures neurological functioning, that's going to cost a lot more than a paper-based survey. So you have to weigh the benefits and the drawbacks also based on your financial constraints. Now let's shift gears. We talked about recruiting a sample. We talked about the independent variable. Now let's talk about the dependent variable. And you may recall the independent variable was the variable that you manipulated. The dependent variable was the variable that you measured. So when measuring the dependent variable, you could do it in different ways, right? So self-report measures. So this is where a person fills out some form of a rating scale, talks about how they felt. Now, it could also be done with open-ended questions to share one's perspective. Or it could be done on a Likert scale, like the LaBranche breast exam study that we talked about. It was competence on a 1 to 7 point scale. So that has to do with rating scales and surveys. You could also measure the dependent variable using behavioral measures. So directly observing a behavior. Now, if you're going to use a behavioral measure, you could observe and take score many different ways. The first way is to say, well, did the behavior occur, yes or no? Well, that's sort of like hit or miss. There are more sophisticated ways to do it, which is the number of times a behavior occurred divided by the amount of time, right? So that would be the rate of the behavior. You could also measure reaction time, or the amount of time it takes before you start to do a behavior, or duration, which is the amount of time from when you start the behavior to when you end a given behavior. So you might use duration and reaction time for tantrum behavior or oppositional defiant behavior, things like that. Okay. In addition, we could also measure from a psychophysiological point of view. One measure is the Galvanic Skin Response that measures sweating. Well, sweating could indicate a whole bunch of different things. Like when we feel anxious, we sweat more. Another measure is the EMG, or electromyogram, and that measures muscle tension. It's interesting, many of you probably have heard of the lie detector test. Well, the lie detector test uses galvanic skin responses, muscular tension, and a neurological firing of the brain, or EEG. So number of action potentials or firing of the brain. But there are more than those three. You could also get information using blood and urine analysis. So you could identify things like diabetes through blood and urine analysis. You could also, if you're looking for a psychological disorder to measure, you could measure substance use disorders through blood and urine analysis. If you're on certain medications, they might want certain blood levels to be taken to see if you're within normal limits. So blood and urine analysis could also be used for psychological purposes. And then we have the MRI and the CAT scan. So the MRI and the CAT scan, which is not on this slide, they are referred to as static imaging devices. Static means still. It's a non-moving picture of the brain. So what's the difference between a CAT scan and an MRI other than how you get the picture? It's resolution. MRIs tend to have greater resolution than CAT scans or CT scans, right? But believe it or not, getting a picture of the brain, even a still picture of the brain, you could identify a whole host of psychological conditions. For example, you might be able to identify schizophrenia. How would you do that? If you have a picture of the brain and it's a coronal slice that's basically going top down at the middle, you could look at the lateral ventricles, and if they're wider than they're supposed to be, that might indicate schizophrenia. You could identify Alzheimer's disease by the hippocampus. It's called a volumetry study, being smaller than average size, and so forth. I could keep going on, but MRIs and CAT scans give you a picture of the brain and potential anatomical changes in the structure of the brain. Then we have the difference between structure and function. So we have the MRI, and then we have a functional MRI. Now we're measuring brain activity while doing a task. So you might have seen those pictures of the brain and they glow different colors, red, green, yellow, and so forth. Well, that's the amount of neuronal activity happening while you do a task. So instead of it being a still image, it's going to constantly be in flux while you do the task. All right, so we talked about having sensitivity on the independent variable, but we also want to have sensitivity on the dependent variable. So as I said, you want to start with the independent variable being very extremely different, but on a dependent variable, you also want to have breadth of sensitivity. So the more response options a person has on a survey, the better the sensitivity. Now, it's possible that when you do a psychological test, you can have a ceiling effect or a floor effect. When you think of ceiling, think very high. So it's possible that the way you design the study, everybody scored or performed really well. That means the task was too easy. So if I give you a test and it's too easy, everyone's going to get it 90s and above. That's called a ceiling effect. The opposite is also true. It's possible to have a floor effect where a task is either too hard or irrelevant to what you're trying to look at, and everyone performs very poorly. So imagine I gave you a test and everyone was getting in the 30s and the 40s. Well, that's not a very good exam. That's not a good measure of your true ability. The best dependent variables get a wide spread of scores. So if you look at tests, you want tests where there are people getting A's, B's, C's, D's, and F's, and the center of the distribution should be C's, right? And then it spreads out. That would be a very good sensitive measure. But if everybody's getting 90s or above, that's what we call a ceiling effect. If everyone's getting 30s or below, that's a floor effect. And if you don't have sensitivity to your dependent variable, you're not likely to find statistically significant differences. So that's sensitivity. The next thing you want to think about is the importance of having multiple measures, having multiple dependent variables, or multiple ways to measure the same dependent variable. So the more things, the more ways you collect data on the dependent variable, if the results are similar over and over and over, so you have the same effect or similar effect using different measures, then your confidence in the outcome increases. Whereas it's possible sometimes they don't match. But the one challenge you have, as I talked to you last lecture, was when you have multiple measures, you have to worry about the order of presentation. You may recall I talked about order effects and the importance of counterbalancing to address order effects. One way to get rid of order effects is run multiple experiments. Who said everyone has to be in the same experiment? You could do experiment 1, experiment 2, experiment 3, and if all 3 experiments come up with the same outcome, once again, your confidence is increased. The same way you have to worry about the cost of the manipulation, you have to worry about the cost of the measure or the dependent variable. So it's cheaper to measure dependent variable with paper and pencil self-reports. But if your dependent variable requires you to pay for multiple observers, to train multiple observers, to buy equipment, whether they be video cameras or EEG machines, that's going to cost you a lot more money. And again, you have to weigh, well, what is your budget? So always thinking about the practical costs. Last but not least, when we do research, we have to worry about ethics and make sure that whatever data we collect on the dependent variable, we protect the privacy and confidentiality. So other things we need to talk about are additional controls. So you may recall that a basic experiment has a minimum of 2 levels or 2 groups. You have the experimental group that receives the manipulation, the control group that receives nothing and serves as a comparison. So it doesn't receive the manipulation. So it's very, very important to make sure you have this, because if you want to say A cause B, you need to have a comparison group. You also want to control confounds to get rid of any possible alternative explanations. All of this should sound familiar from lecture 8. Now, in terms of reducing response biases, we have to worry about participant expectations and experimenter expectations. So these could bias your results. So let's talk about participant expectations. So remember how I said that sometimes you might want to disguise the purpose of your study? And the reason is because there's something called demand characteristics. And that is, it's almost as if you demand something of the participant. If a participant figures out what the purpose of your study is, more often than not, they're going to behave in accordance with your hypothesis rather than oppose it. So you might get the results you want, not because they're the true results, but people wanted to please you as a participant. So if you think that that's absurd, here's some old research that's been supported time and time again, but the early research of Urine demonstrated people are cooperative. So let's say I had you volunteer for a study, and it had a bunch of math problems on it, and you had to add up numbers on a worksheet. And it wasn't very complicated work, so you should be able to breeze through the, let's say, 15 problems on the page. Well, at the bottom of the page, it says, when completed, bring it up to the front of the room and pick up a card for more instructions. So you pick up a card, and the card says to fold the paper up and tear it into 32 pieces, and then take a new page, pick up a new page. So you do that next page with 15 easy math problems of addition. And at the bottom, it says, go up to the front of the room and pick up a card. And what does the card say again? Fold it up and tear it into 32 pieces and take another page. Well, how many times would you need that set of instructions for you to figure out that it's going to keep telling you to do more math problems, and then when you pick up a card, it's going to keep telling you to rip it up? Maybe 2 or 3 times. So if you're doing basic mathematics, you should be up there every 5-10 minutes. What if I told you that participants would spend hours doing this math problem and ripping up their work, hours without protest? That's absurd. They should know that the next card's going to tell them to do more work. That is bizarre. So what did Orrin interpret? Orrin said, well, guess what? They're just being cooperative. If a researcher tells you to do something, more often than not, you're going to be cooperative, even if you know that it's busy work or not useful. So if you're worried about demand characteristics or people figuring out your hypothesis and behaving in accordance with it, one way to do this is use deception, which we talked about. And deception comes in many ways, either to misrepresent what the purpose of the study is, or to have some filler items or questions on the experiment that have nothing to do with your hypothesis. And the whole reason why those questions are there is just to distract the participant so that they don't figure out the true nature of the study. So another thing you could do is assess demand characteristics. How would you do that? You could say, thank you for participating in my study. What do you think the goal of the study was? Well, you could take note of that. If they said, well, in the previous Orange study, you were just trying to see how cooperative I was, then you would say, yeah, they figured out my hypothesis. And then you could separately analyze those who accurately or inaccurately guess the hypothesis to see if there are any differences. Now, if it's not a questionnaire or it's not an experiment that you're observing them directly, another way to do it is to use unobtrusive measures. So now you're observing a person's behavior without their knowledge. And the purpose of that is that if they don't know you're watching them or collecting data, they're going to behave more naturally or normally. And that gets rid of demand characteristics. So one form of demand characteristics, which we haven't talked about that much, but I alluded to in the last lecture, was something called a placebo effect. I want you to imagine for the time being that you are a medical doctor and you have a patient that keeps coming into the office and they say, Doc, you know, I have these migraines and they just won't go away. So you do neurological assessment. There's nothing neurologically wrong with them. So you know that it's not really anything going on. So you say, oh, nothing's going on. You send them away. They come back a week later and say, oh, Doc, I have these stomach problems. I don't know what's going on. So you do a gastrointestinal workup and you find out that there's nothing wrong with them. So you tell them, okay, nothing doing here, go home, blah, blah, blah. after coming to your office repeatedly, you're going to formulate that maybe there's a psychological condition going on here called hypochondriasis. So you might do something like House did in Gregory House did in the show House, buying candy and putting it in a pill jar. And so you might say, here's some medicine. I know you're struggling. Take one a day. I'm going to check in on you. And so the person takes these sugar pills, which is a placebo. And on day one, you call them and say, how are you feeling? And they're like, well, doc, I'm feeling a little bit better, but still, I have these problems. It's not fully resolved. Okay, keep taking the pills, you tell them. And then a couple days later, you call up, how are you feeling? Oh, doc, I'm starting to feel better. And so as a doctor, you're like, that's really great. Keep doing what you're doing. Then you call them another time at the end of the week, and they say, wow, doc, I don't know what you gave me, but for my migraines and my stomach problems, they've gone away. So at that point, you say, once you come to my office, you bring them in and you show them the Pez dispenser. And you say, well, actually, the pills that you were taking were candy. They weren't actually medicine. And why is it that that person who had hypochondriasis actually felt better? The answer is placebo effect. If you go to a doctor and the doctor prescribes medication, you expect the medication to have a desirable outcome for whatever it was prescribed for. So that person who felt better, it was all psychological. And that comes not just with the extreme example that I gave, but with all medication. We have to be aware that it's possible that there's some form of expectancy on the participant that they start to get better on their own. So if you're creating a drug to treat a condition, you have to have placebo groups to be able to separate how much of the change in your psychological well-being was due to taking the real medication or was due to taking this placebo, and it was all placebo effect. So you want to measure that. And if it was a meaningful effect, you should have a very small placebo effect and a large real effect. But the larger the placebo effect, the less confidence you have in that medication. Here's another example. So Marla and Rosenau did a study on alcohol and expectancy. So group one was told that they weren't receiving alcohol and they didn't receive alcohol, right? So their expectations and what they got was it matched. So are they going to psychologically be impacted by something? So let's talk about it more specifically. You could give someone Coors as a beer, or you could give someone O'Doul's as a non-alcoholic beer. So in group one, they were told this was O'Doul's or non-alcoholic, and that was that. Now in group two, they were told they were not receiving alcohol, right? And then they actually did receive alcohol. Group three, they were told that they were receiving alcohol, so they expected the Coors, but you gave them the O'Doul's. So should they have any changes in their behavior due to alcohol in group three? No, right? Because they didn't actually receive alcohol. And then in group four, they received alcohol and they expected alcohol. What was remarkable was that group 3 and 4 responded very similarly in this study, even though in group 3, they didn't receive alcohol. Why is that? The answer is simple. For those people who drink alcohol, they know how alcohol is supposed to affect them psychologically. They're supposed to become more uninhibited. So group 3, because they expected to get buzzed or drunk, whatever it might be, their behavior changed as if they were buzzed or drunk. Another example of expectancy on the participant. So when using a placebo control, it's very important to understand the ethics around it. Why do we use placebo controls? To test out how much is placebo effect. But what happens if you find a real difference? And I see someone raise their hand. I'll take questions at the end. If you see that it had a meaningful difference, you're supposed to give the control group the drug at the end of the trial. And the reason is that when people volunteer, they're told that you could be receiving the placebo or the real thing. But everyone who volunteers wants to be in the experimental group. They want to receive the treatment. So ethically, if it works, you should treat the people in the control group at the conclusion of the experiment. So that's the participant and worrying about your participant and reactions from your participants. We also have to worry about experimenter biases, right? So an experimenter bias or an expectancy effect on this could be the experimenter either intentionally or unintentionally influences the participants. How might experimenter bias work? Either you treat the groups differently, and that could happen intentionally or unintentionally, or when you're scoring the results, you score people differently based on your expectations. So for this, I'll give you a history of psychology example. So there was this famous study in Germany where they had this horse that seemed to be very, very brilliant, an intelligent horse that could add, subtract, do multiplication, division, did memory, and it answered questions by tapping and nodding its head. And its owner, Wilhelm van Oosten, was like, see, animals have intelligence. Animals could do cognitive processes. And Wilhelm van Oosten wanted to argue that if the only difference between humans and animals are that humans have a formal education process. So Stumpf, which was one of the German Gestalt phenomenologists at the time, he was asked to do an investigation to see if von Oosten was a fraud. So he went and observed, and Stumpf could not find any fraud or deceit. But Stumpf had another student by the name of Funkst and said, I can't figure this out, but can you see if there's anything going? So Funkst had a genius idea. What Funkst did was he designed an experiment where the von Oosten knew the answer and where von Oosten didn't know the answer. And what he found was that clever Hans, the horse, got the answer correct only when the owner or the questioner or the group knew the answer. But if they didn't know the answer, Hans, the horse, didn't get it right. So what was happening? There were these unconscious body language that the questioner was giving. So what he found, Funkst found, was that people would look up in the audience when Hans was supposed to stop. And Hans, the horse, read the cues and stopped tapping. So if they didn't have the answer, they couldn't do that. So what did that tell you is that sometimes the audience, sometimes the experimenter, could give off cues that could skew the results. So that's one thing on an unconscious level. So that would be the examiner metaphorically treating the people differently, or how that could work. But you could also have a similar outcome in scoring purposes. So Rosenthal did another example with his grad students. And he told his grad students that he had a bunch of mice that were in two different groups. The maze bright groups were considered really smart. The maze dull mice were considered really dumb or unintelligent. And he had his students measure the amount of time it took for the mice in group 1 versus the mice in group 2 to successfully navigate a maze. Now, what did he find? His graduate students reported out that the mice who were considered maze bright solved the maze much quicker than the maze dull mice. Except there was only one problem. Rosenthal took these mice from the same litter. There was nothing more intelligent about one group of the mice over the other. But merely putting a label on the mice, the graduate students treated preferentially the maze bright mice over the maze dull mice. How could that happen? It could be either that they artificially manufactured the time of completion, or they might have stopped the clock prematurely, thinking that the maze bright mice solved the puzzle.
Professor?
Yeah.
Sorry for interrupting, but this is important. I sent a message privately through the Zoom chat. However, whenever you can, could you please look at it? I do have to leave this Zoom meeting though, because I have something important that I have to attend to. But it's just regarding the assignment that's due this Friday. So if it's possible, could you send me
Yep.
a Zoom link for tomorrow office hours?
Gotcha. Sure.
What time are your office hours?
It's on the syllabus.
Okay. Are you gonna send me the Zoom link through my email?
It'll be on the, I'll put it on Canvas.
Okay, please. Thank you.
You got it. Take care. All right. So, yeah, so where was I? Sorry, Brittany. Let me think. Okay. So, the application of this study. So, the application of this study, it is also when we label people. If you look in the school system, people are labeled, or they're tracked as gifted and talented, or people might be labeled as these people are ready to blossom. Well, you could unconsciously give them better scores or give them more opportunity based on that. And we see in education that does happen. It's a concern. All right, so how do you deal with expectancy effects, right? So how do you deal with that? Well, there are different ways you could do it. One, if you're worried about treating your participants differently, run all of your participants all at the same time. Because whatever instructions they get, everybody gets. Or you could train the experimenters to go through a script, or you could automate the procedures, or you could use single and double blind experimental procedures. A single blind experiment is where the participants don't know the hypothesis. A double blind experiment is where two groups of individuals don't know it, hence double. Both the participant and the experimenter don't know the hypothesis, which begs the question, how is it possible that an experimenter may not know the hypothesis? Well, one way is to have two sets of experimenters. One set of experimenters who designs the study, and a second set of experimenters who collect the data. So the people who design the study know the hypothesis, but they can't artificially score it because they're not doing the data collection. And the people doing the data collection, they don't know who's in what group, so they can't manufacture the results either. Other things you want to consider: research proposals. Research proposals are highly important. They give you an opportunity to review the literature. Proposals are useful when submitting an IRB application or grants, but they also give you an opportunity for feedback from your colleagues. They give you opportunity to spend time thinking about data collection before it's actually due. So you want to consider proposals. You also might want to consider pilot studies, because there are a lot of studies you might spend years doing and find that the results were not significant. And if that happens, you may feel like you wasted your time. So one way to see if anything is going on is something called a pilot study. And a pilot study is a shorter or smaller scale version of the study, just to see if your manipulation is effective, just to see if people understood the experiment, just to see if people had questions or feedback that they had for you to refine your hypothesis. But all of that gives you an opportunity before spending a lot of time to make modification. So we also have manipulation check. I said to you at first, the first thing you want to do is have your two levels of your dependent variable extremely different. So you want to see if your independent variable causes changes to your dependent variable. If you have very different groups, so let's say you had a similarity score in relationships of a 1 versus 10, and you didn't find any meaningful pattern, then at that extreme difference, your study's a dud. So you might want to do a manipulation check to see if your independent variable is even trending in the direction you want. So it saves you money, saves you time. It also gets you thinking about what happens when you have non-significant results. Now, we talked about deception. If you have deception, you can do debriefing, which is opportunity to talk about the true nature of the study. It adds value. People can offer feedback. And the decision when to do debriefing is one that's personal. You have to decide whether people are going to talk to one another. If you feel that people are going to talk to one another and influence your results, then you might wait till the end of data collection to do debriefing versus right after they participate. And then after you do data collection, you do data analysis. And we'll talk about statistical tests in chapter 13, but statistics are your way of identifying patterns of results to see if there's really some kind of relationship between your independent variable and your dependent variable. So if you want to say your independent variable caused changes to your dependent variable, the only way to do that is to do a statistical analysis. So now you've collected your data. Now you've analyzed your data. Let's assume you found meaningful differences. Now you want to communicate your data. Now you want to get your results out there. There are different ways to do this. The first of which is professional meetings. So most nonprofits have annual meetings. Most psychological societies have annual meetings. So the American Psychological Association has an annual meeting. You saw New York State Psychological Association has an annual meeting. Many different groups have annual meetings. So you could submit your work to provide a lecture, a poster, or some form of an oral presentation at a professional meeting. That's one way to do it. And then the second way to do it is to write it up and try and get it published in journal articles. Now, you may recall I said that when you submit the journal articles, you have several of your peers reviewing the results to identify potential flaws or methodological issues. And what we find is that 9 out of 10 times, people who submit get rejected, and they're offered meaningful feedback. They work on it. Either they resubmit it to the same journal or another journal, but eventually it gets published. And that is our lesson. So I'm going to stop share, and I'm going to stop the recording, and I'm going to see